Methylene blue as an alternative electron carrier for the oxidative phosphorylation process by cancer cells. What is meant by this?
The idea of methylene blue, MB, acting as an alternative electron carrier and its role in restoring oxidative phosphorylation in cancer cells is rooted in the unique metabolic behavior of cancer cells and the biochemical properties of methylene blue. Let's break this down step by step. 1. Cancer cells and the Warburg effect. Cancer cells often rely on glycolysis, fermentation, for energy production, even in the presence of sufficient oxygen. This is known as the Warburg effect. Instead of using the highly efficient oxidative phosphorylation pathway in mitochondria, cancer cells ferment glucose into lactate, which is less efficient but allows them to survive in low-oxygen environments and rapidly proliferate. This metabolic shift is often due to mitochondrial dysfunction in cancer cells, where the electron transport chain is impaired, preventing normal oxidative phosphorylation. 2. Methylene blue as an alternative electron carrier. Methylene blue is a redox-active compound, meaning it can accept and donate electrons. This property allows it to act as an alternative electron carrier in the mitochondrial electron transport chain. In healthy cells, electrons are passed through a series of protein complexes, 1, 2, 3, and 4, in the mitochondria to generate ATP. In cancer cells, these complexes, especially complex iron 3, are often dysfunctional, disrupting the flow of electrons and reducing ATP production. Methylene blue can bypass these dysfunctional complexes by directly shuttling electrons to cytochrome C, a key component of the electron transport chain, effectively jump-starting the electron transport chain and restoring ATP production through oxidative phosphorylation. 3. Restoring oxidative phosphorylation in cancer cells. By acting as an alternative electron carrier, methylene blue helps cancer cells revert to oxidative phosphorylation instead of relying on glycolysis. This shift has several potential effects. Reduced glycolysis. Cancer cells may produce less lactate, which can alter the tumor microenvironment and slow tumor growth. Increased ATP production. Oxidative phosphorylation is more efficient than glycolysis, but cancer cells are often poorly adapted to handle the increased oxidative stress and energy demands of this pathway. Induction of apoptosis. Restoring mitochondrial function can trigger programmed cell death, apoptosis, in cancer cells, as they may not be able to cope with the restored metabolic activity and increased reactive oxygen species, ROS, production. 4. Why this matters for cancer therapy. Cancer cells' reliance on glycolysis is a key survival mechanism, allowing them to thrive in low-oxygen environments and resist apoptosis. By forcing cancer cells to switch back to oxidative phosphorylation, methylene blue disrupts their metabolic adaptation, potentially making them more vulnerable to treatment. This approach aligns with the metabolic theory of cancer, which suggests that targeting the unique metabolic vulnerabilities of cancer cells could be an effective therapeutic strategy. 5. Practical implications. Methylene blue as a therapeutic agent. In preclinical studies, methylene blue has shown promise in reducing tumor growth and enhancing the effects of other cancer treatments. Combination therapy. MB could be used alongside other therapies, e.g., chemotherapy or radiation, to exploit the metabolic vulnerabilities of cancer cells. Challenges. While the concept is promising, more research is needed to determine the optimal dosage, delivery methods, and potential side effects of using methylene blue in cancer therapy. Summary. When we say methylene blue acts as an alternative electron carrier and restores oxidative phosphorylation in cancer cells, we mean that it bypasses dysfunctional parts of the mitochondrial electron transport chain, allowing cancer cells to resume normal energy production. This metabolic shift can disrupt the cancer cell's survival mechanisms, potentially leading to reduced tumor growth and increased susceptibility to apoptosis. This approach represents a novel way to target the metabolic weaknesses of cancer cells, offering a promising avenue for future therapies.